Hello, everyone. My name is Alisher Talgatov. I'm a graduate student at Dr. Shri McFarland's research group here at UTA, and it is my pleasure to present our work called Photobiological Applications of Light-Activated Metallic Drugs. Our research group focuses on studying photodynamic therapy, or PDT. So, how does it work? A person with cancer receives the drug called photosynthesizer, which is being absorbed by the cancer cells throughout a certain period of time, and once exposed to light, it generates reactive molecular species that are killing cancer cells. Our research group has developed and synthesized TLD-1433, or Ruvidar, which is the photosynthesizer based on the metal, ruthenium polypyridyl oligotinyl complex. It is currently undergoing phase 2 clinical trials against non-muscle invasive bladder cancer, and among 90 patients enrolled, 78 already been treated and 12 pending study completion. To understand the properties of TLD-1433, we established structure-activity relationships where we could modify the ligand environment and understand the photophysical, photochemical, photobiological, and other properties. So the purpose of this work was to synthesize, characterize, and evaluate ruthenium bis terpyridyl complexes, starting with the reference compound ruterp2, and adding some conjugation via appending a ligotinyl chain starting with 1 and ended up with 4, and elucidate how this conjugation would modulate photophysical behavior and photobiological activity. In terms of synthesis, to get the reference compound, we need to react ruthenium trichloride with terpyridine in 1:2 ratio. For the rest of the complexes, we needed to perform same reaction but 1:1 ratio to yield the intermediate, which were further reacted in situ with the terp-NT ligand synthesized separately in the TA, and then we get our desired complexes isolated and purified using the flash chromatography. To confirm our structures of the complexes, we performed extensive NMR studies using 1D and 2D techniques in consultation with literature, assigning all the hydrogens for every molecule of the series. And we further ran fluorine NMR spectrum to confirm all of the PF6 ions were removed after purification. So our complexes were studied photophysically, where we highlight here the steady state UV absorption, having two features which corresponds to the one IL-ILCT transitions here, corresponding to the transitions within the ligotinyl chain, and MLCT bands standing with the pi star transitions from the metal to the ligand, both increasing in intensity upon tyrosine addition, and we observed the redshift or batachromic shift for both of those features. To understand the excited states of the molecules, we performed the transient absorption spectroscopy and the emission spectroscopy. For the first one, we observed the bleaching for all compounds, whereas compounds with a larger number of tyrosines, we observed the population of our new excited state, which corresponds to the oligotinyl addition. For the emission spectrum, we have observed the same red shifts and increase in the lifetime, which indicates that expansion of the pi system using the tyrosines tunes the both energy and configuration of the emissive states that are responsible for the generation of molecular species. So we further confirmed with our photobiological evaluation by getting the activity against melanoma cells, where our complex with two tyrosines was pretty active in under green, blue, and visible light under normal oxygen conditions, and ruthenium with four tyrosines was also active in red light, which indicates the wavelength and oxygen-dependent structure-activity relationships. And to sum up all the work performed, we synthesized a new family of bisterpyryl oligotinyl complexes, and we achieved the triplet ILCT state to get a higher photobiological activity on the tyrosine addition. And once we increase the number of tyrosines, it energetically positions the triplet ILCT state to be accessible upon photoexcitation. And I would like to thank all the funding, Department of Chemistry and Biochemistry. If you're interested in our work, check our website, social media. And if you have any questions, feel free to reach out to me personally or through group. And with that, I want to wrap up our poster. I want to thank Discover organizers for having this project running in a yearly basis. And let's celebrate 60 years of the UTA College of Science. Thank you.